Welcome back to the London Futurist podcast. If you like the show, please consider leaving us a 5-star review on your podcast platform. It helps others to find us. And feel free also to send us questions and suggestions. Also, we're looking for sponsors to fund a marketing initiative to help us reach a wider audience. The future is rushing towards us, and the better informed we all are, the better it will be. Alex Zhavoronkov is our first guest to make a repeat appearance on the London Futurists podcast, having first joined us in episode 12 last November. David and I are delighted to welcome him back because he's doing some of the most important work on the planet, and he has some important news. As some of you will remember, in 2014, Alex founded Insilico Medicine, a drug discovery company which uses artificial intelligence to identify novel targets and novel molecules for pharmaceutical companies. Insilico now has drugs designed with AI in human clinical trials, and it's one of a number of companies that are demonstrating that developing drugs with AI can cut the time and money involved in the process by as much as 90%. So Alex, welcome back to the London Futurists podcast.
Thank you, Callum. Thank you, David. Very happy to be with you.
Thanks for joining us again, Alex.
Alex, let's start with your exciting announcement, which is hot off the press. What milestone has Insilico Medicine recently announced?
Sure. Very happy to announce that now our AI-discovered, AI-generated molecule has entered phase 2 human clinical trials for idiopathic pulmonary fibrosis, an age-associated disease, which is pretty deadly and is related to many other age-associated diseases, according to our studies. So we've managed to discover a novel target utilizing a multifactorial, multimodal AI, which utilizes biological data and text data, utilizing many different AI models that provided a consensus for several targets that are implicated in aging and fibrosis at the same time. We've done that in 2019. So again, it's not something that we've done overnight. After that, we validated a bunch of targets that resulted from that early experiment. We prioritized 20 targets, managed to test 5. All of them worked. Then we prioritized 1. And then we decided to go after this target with our own chemistry. So for those of you who are not familiar with the drug discovery process, most of the companies in our field in AI-powered drug discovery are focused on old targets that are very well known, very well characterized, and they just try to design small molecules or antibodies utilizing artificial intelligence. And usually that's not exactly generative AI that people are using, at least that's not GANs or transformers.
A target isn't a disease. A target's a part of a complicated biochemical reaction pathway. Is that correct?
That's correct. So first you formulate the mechanism of disease. So you try to formulate your disease hypothesis, and then you narrow it down to a set of proteins that we refer to as targets that later we are trying to drug, meaning that we are trying to disable those proteins or activate them in order to achieve the disease-modifying effect. So the first step is always biology. You figure out why the disease happens and what's driving it, narrow it down to one protein or several proteins in a pathway. And after that, you design the small molecule chemistry, so a drug that would go after that target, or an antibody. In our case, we're a small molecule company, so we decided to go after this target of interest with a small molecule, meaning a tablet that you can swallow and get those molecules into your bloodstream. And we used a form of generative AI that allows you to, instead of searching for a needle in a haystack, trying to find a molecule out of the many known ones, out of the many molecular libraries, we decided instead of doing that to imagine novel molecules with the desired properties. So now it's actually much easier to explain this year because the concept of generative AI was not exactly understood by the general public the last time we spoke. But we were in generative since 2016, when my first paper on the applications of generative adversarial networks to small molecule design was published. And then we started using more complex architectures and then transformer neural networks. and then developed software that now is used by almost half the pharmaceutical companies. And we used that software that imagines novel needles with the desired properties to target this protein target that we've managed to identify using a different form of AI.
Alex, you're one of the first people I ever heard the word transformer AI from. So I'd like to dig into that a little bit because many of our audience will be aware broadly that this new type of AI has arrived called generative AI, but they won't be all that clear exactly what it is. Back in 2012, there was a big bang in AI when deep learning arrived. And then in 2017, Google researchers published a paper called All You Need Is Attention, and that ushered in this new wave of AI called transformer AIs. The deep learning revolution gave us AIs which classify, they discriminate between things. And the generative AIs do what it says on the tin. They generate text, they generate images, and it's incredibly powerful, as we've seen since the launch of GPT-4. But as I said, you were talking about it when we spoke last time. How exactly are you using transformers and GANs, generative adversarial networks, which is sort of an older form of generative AI? How are you using them? What difference has that made to your processes? Because obviously you didn't start out using them, you adopted them when they came along.
Transformers are very well known because of GPT, ChatGPT, GPT-3, GPT-4, and GPT stands for Generative Pre-trained Transformer. What makes that technology powerful is exactly the attention layer that you've talked about that was published in 2017 by Google researchers. That allows you to be able to predict the next token in a sentence, the next sentence in the abstract, or the next abstract in the book. That's why those large language models are so powerful. I wouldn't say that generative adversarial networks are significantly older. They were published 2014 and are employed abundantly in many different applications. So I would say it's a competing form of generative AI. It's also rather computationally intensive and data intensive, just like transformers. But for GANs, you really need to use very large architectures and large amounts of data and training to achieve high level of accuracy. So we used GANs since 2015 and started publishing 2016. A GAN is an architecture where you have a generator, a deep neural network that generates meaningful data in response to some input that you give it. For example, some desired properties. It could be text, it could be specific properties of the object that you are trying to get. Like, for example, we started utilizing those generators to generate human biological data, human biological profiles of people of the desired age very early in the process. In 2017, we applied for our first patent on that. So generation of human biological data with age as a generation condition. That's where actually aging research comes very useful. So the generator generates meaningful data, and another network called the discriminator tries to evaluate whether the output of the generator is true or false. So it's kind of a policeman. One network is trying to trick another into believing that the data is ground truth, and another one is evaluating whether it is close to ground truth or not. In the process of this adversarial competition, generation and discrimination, those two networks learn to generate really amazing high-quality data. By the way, this technology can be very easily combined with transformers, with large language models, with attention. Usually when we develop our models, we usually use many different generative systems that generate molecules with the desired properties. So you give those models a crystal structure, the structure of the protein of interest, the target. You sometimes give it also some molecule that we know may bind to this target already to some pocket. You annotate additional pockets where those molecules can bind. And after that, you give this crystal to this ensemble of generative models, and all of them start generating molecules that actually fit your generation conditions. However, unlike with ChatGPT, we are not interested in snappy response. I don't need to see those molecules right away because I have many months or years ahead of me to validate the accuracy of those models. So what I do, I have a huge number of predictive models that look at each individual molecule and predict whether it has the property we're looking for or not. So I have more than 500 of those predictive models. So think of it as an ensemble of generators trying to generate many, many different molecules. And I've got a filter which basically evaluates each individual molecule and then rewards or punishes each model that created the molecule based on whether the property is there or not. And we run this process for 72 hours usually. Sometimes it's longer, sometimes it's shorter, depending on the complexity of the protein. And usually after 72 hours, those pre-trained models, some of them are transformers, some of them are GANs, some of them are genetic algorithms, some of them are variational autoencoders. So there are multiple generative architectures. And actually, from scratch, you don't know which one is going to perform better for your reward objective and for your desired properties for your protein of interest. So they all are good, usually. You don't know which one is going to outperform.
And you haven't found that one type tends to work better than other types? They're all equally capable of producing good results?
We'll be right back after a quick break.
The People in Performance podcast offers insights into the strategic competencies, behaviors, and results needed to increase the performance of employees and leaders. The show looks at all areas of the world of work, including attraction and recruitment, onboarding, training, and succession planning. Tune in to get a better handle on the stages of workforce planning you need to master in order to guarantee long-term success in a rapidly changing world of work. New episodes drop each week. Check out the latest People in Performance episodes on your podcast platform of choice. Follow us on socials and read the latest articles at pppod.com.
Some are better for some protein classes, and we usually know that. But we usually give other models a chance because for us, we also value diversity. Diversity in chemistry is extremely important and novelty. And very often, some models can produce really great chemistry that satisfies your desired conditions, but the molecules are not going to be super novel or super diverse. You really want to have diverse scaffolds. Just like in any organization or an ecosystem, diversity is very important. So in chemistry also, when you get a range of original molecular scaffolds that you want to satisfy your requirements, you want to have large numbers of very different molecules. But you also want to ensure that each one has the properties you need. So you've got a lot of different needles, but some of those needles may be Aztec style, or some of them may be super futuristic, and some of them may have 5 different needles. Some of them are ninja star needles. So you want to have many, many different options to select from. And after 72 hours, many of those models that are warmed up with the reinforcement learning ensemble, usually they start producing really good molecules. Then you give each model a chance. You generate, let's say, 100. Then the professional medicinal chemist would look at those molecules, look at the predicted properties, and say, okay, I have the budget to synthesize, let's say, 15 or 20. Usually some of those molecules work, but they are still not super perfect. And you then feed those molecules that worked as templates into Chemistry 42, into this ensemble, again with a crystal structure, and do this one more time.
And that part where you're testing these 15 or so examples that you've got, that's all done in the real world, so to speak, in wet labs. That's not done in simulations.
Yeah, absolutely. We rely on the power of many contract research organizations. We usually work with around 40, where you can order chemistry to be made and then very rapidly tested in biological assays. So we follow kind of the Apple model, designed anywhere in the world, but tested in many contract research organizations. Usually it's China or India. where this infrastructure is extremely well developed and you've got hundreds of thousands of professional chemists and biologists to very rapidly synthesize and test. And some of those molecules that we produce are easy to synthesize and some are very difficult. So the synthetic route might be very long, but you still want to prosecute those molecules. And that process that I've just described, we've done that in late 2019, early 2020 for the program that we are talking about.
That's really helpful, Alex. That's a really good grounding in how the process works. And now you're at phase 2 human trials for this potential treatment for idiopathic pulmonary fibrosis.
Yeah.
What does phase 2 human trials mean?
That's where the rubber hits the road and you test efficacy in real patients. To explain the process of how we got to this molecule, so yes, we used AI to identify the protein target, fed it into another form of AI with many generative engines to generate a bunch of perfect molecules. Then we had to take those molecules into mice and other animals, and also in many experimental systems to demonstrate that those molecules work in a model of a disease, and also that those molecules are safe. And then we had to take those molecules into dogs, for example, and conduct long-term studies before you touch humans. And then we did phase 0 in 2021. It was actually COVID pandemic everywhere. So we went to Australia, recruited 8 healthy volunteers, got the approval to give them the drug, gave them the drug, studied this drug at length in those healthy volunteers, and then we got the approval to start phase 1 human clinical trial where you test safety at length. And we did that in New Zealand and in China, 2 large studies, and demonstrated that the molecule is safe to proceed with and safe for a chronic indication, because as you can imagine, there are many different safety studies for something that is super acute, like a virus or a bacteria, and something that is as acute as cancer, for example. So you are trying to fight cancer and you can tolerate certain level of toxicity. When it comes to a chronic disease, you're going to be taking the drug for sometimes your entire life. So you need to ensure that your safety profile is significantly different from the drugs that are designed for acute conditions. So we had to demonstrate safety in a large number of healthy volunteers. And now that we've done that, we are giving this drug to real patients with IPF in a phase 2 clinical study in the US and in China. And we already dosed our first patients. That's also a very big milestone. And for those of you who are not familiar with clinical trials and also IPF, it's not an easy trial to run, because usually those patients expire very quickly. They usually were treated using multiple other drugs. They're usually reasonably old, at least significantly older than average age of a cancer patient. Average age of onset is around 60, 65. And it's very important to enroll patients that are likely to benefit and at least get through the study. It's very difficult to select those patients. Also, IPF is not a disease that has very high prevalence. It's rare, but not so rare. It's not as rare as ALS, for example. It takes time to recruit. So right now we're recruiting and they're doing the testing. So it will take a while.
How many patients or test people are you going to be recruiting?
We have not announced that, but it's many. We need to get a large number of patients. You don't recruit them right away. You have to do it over time, and the study is going to take a while. The beauty of this study is something that we have not disclosed openly, but it's pretty obvious. We've also nominated the preclinical candidate for the same target for kidney fibrosis. Kidney fibrosis is something that you and I, we both have. It increases with age, and it's the leading cause of chronic kidney disease. That's 15-16% of the global population. We might not die because of it, but we are likely to die with it. And the kidney function usually diminishes with age pretty dramatically, right? And we get all kinds of problems there. And in significant part is driven by the fibrotic buildup in the kidney. So you get your normal tissue that is performing useful work replaced with fibrotic tissue, which is essentially a patch. So it's your Band-Aid that the body developed to slowly but surely replace the working tissue with those Band-Aids without the need to recycle, because nature did not intend us to live for hundreds of years. It intended us to reproduce, take care of our young, and then decline and die.
So fibrosis, in short, then, is when the body replaces tissue which used to be functioning perfectly well, stopped functioning perfectly well. The body replaces it, but doesn't replace it with tissue which is as good as the original.
Yes.
And it's a bit of a makeshift repair.
Correct. So think about scarring. If you're wounded and you start healing, the first thing you get is fibrotic buildup, and that's why you usually have the scar. So the scar tissue is your fibrotic tissue as well. Usually it's not just fibrotic tissue, it's many, many other things. It's connective tissue that's building up. You've got all kinds of mineralization and many other things, but basically in brief, those are scars. And you get those scars everywhere, in your heart, in your kidney, in your lung, in your skin, everywhere. So fibrosis is a big driver of aging. And if you know me at least a little bit, you probably know that I am religiously committed to aging research.
Yep, we'll definitely come to that because that's a really important part of you and your work. But just before we get there, I want to ask, the work you're doing with IPF, idiopathic pulmonary fibrosis, is a really important test case in the use of AI to develop drugs. It's not the only one, but it's a very important one. And you've made enormous progress. I know that you have been able to get to the stage where you're at with this drug much faster and much more cheaply than the traditional way of developing drugs. What overall has been the saving in terms of time and cost, and where are the main saving areas?
Looking at apples to apples comparison, we probably shaved about a couple years from the discovery and development process, considering the current novelty profile of the target. If the target is not novel and you are just going after chemistry, we have a bunch of studies like that because, we also need to eat, so to speak, hunt, kill, and eat. And we also do something similar commercially where we do those work for the others, or we make the programs for sale, so to speak, in cancer. Pharma doesn't like to get novel targets early, so we also have a few targets that are not exactly novel. And there it takes about a year on average to get to preclinical candidate with new chemistry, maybe a year and a half. But for novel targets, where the biology is poorly understood, where the structure is poorly understood, and you need to do a lot of experimentation, usually the process of going to preclinical candidate may take you 5 years plus. We went from 0 to 1 with this target profile to preclinical candidate in approximately 18 months. And then to phase 1 in approximately 30 months. That's also included phase 0. Usually people don't do that. They don't do that small pilot because in our case, the target is new. We wanted to make sure that we do additional studies. And then it's around 40 months to phase 2. Usually to get to this stage, you would spend about 6-7 years. I would say closer to 6 than 7 using traditional methods. And usually you would fail 99% of the time because of the target choice. So that step is usually performed in academic labs, and pharma usually just does double validation. In our case, we actually discovered the target ourselves and did a lot of work on the biology side to get to this point. So the most important aspect that we focus on is the probability of success. I actually am not trying to cut costs. Very often I spend much more money per experiment than Big Pharma does, because we usually do the work in 2 parallel labs, in 2 different CROs. If one fails, both have to repeat the experiment, sometimes even for free, because it means that somebody screwed up. So it's a very good idea to do redundant studies because it becomes reproducible. And then we always try to go after many more indications. That's why we also nominated the preclinical candidate for the same target for kidney fibrosis. We also did a lot of work in skin fibrosis. We did a lot of work in liver fibrosis. I don't want to talk about those because even though we demonstrated really, really amazing efficacy in liver fibrosis, it's not a super commercially tractable disease indication, and investors don't like companies going after NASH, for example, nowadays, because there were so many failures. And it's such a multifactorial process, very often driven by metabolism. People just overeat. Those are obese people, don't exercise, etc. And the drugs that target fibrosis may not always help. We try to do many more experiments than usual. So that's costly, but because we did not fail many times, it's actually much cheaper.
So if I understand you right, you've probably saved around about half the time in the process, which is a hell of a saving. The cost maybe is a net, washes out. But the real saving is cutting down that 99% failure rate. That's the real saving.
That's correct.
Presumably, other companies must be very interested in what you're doing. I think you mentioned previously that Big Pharma is quite interested, but they're also cautious because it means changing many of their venerable, time-honored processes. Are they paying more attention now, given that this drug candidate seems so promising?
Oh, yeah. So it's not only that. One other thing that just transpired in our industry, the industry has consolidated dramatically. When we last talked, there were probably a couple hundred companies out there doing something similar.
When you say that, you mean companies applying AI to drug development?
Yes. And they were all buzzing, producing all kinds of news. Sometimes those companies were built by investors just to access financial markets. It's not unusual for an investor, actually it's more common than you think, to understand that there is a trend, to look at a company like us. Usually they all looked at us a little bit and they realized, okay, well, I can take a small piece of Insilico and play a long game because we did not really focus on immediate financial gain. We really wanted to go 9 yards. We're here for the long run. But they realize, okay, well, there is a window of opportunity to go public with those companies. And what they did is they bring, for example, a really famous professor or very famous drug hunter to say, okay, well, this is the founder. They bring a few people from the AI community to spice it up. Usually those are big names. And basically try to assemble the football team for this thematic trend, which is AI. Usually they start with nothing, so they did not really have anything working. They are trying to build this team to build, and then they give them a hell of a lot of money. Sometimes it's 100 million, 200 million, 300 million, and they give them a story that is appealing to generalists or pension funds, family offices, etc. So sometimes that story might be, okay, there is not enough data, so let's generate a lot of great high quality data or buy clinical data, and then we're going to in-license the drugs or buy some companies that will allow us to make those drugs faster. And those companies started mushrooming the industry because, as you remember, 2020, 2021, COVID hit. There was a mega investment into biotech. We went through a hype cycle, and a lot of those companies got the money, but they don't have the tech. And now pharma companies start realizing that, investors start to realize that, and if you don't have the really good tech, and the investor and you're public, or if you're private and you did not demonstrate significant progress, they now see it. Because now the level of competence in the investor community and also even in the media became much more competent. And they understand that, okay, well, now many of those companies do not have the tech. They are there. They have some cash. Some of them didn't have cash. And just recently, you've seen that, for example, Cyclica and Valence, they were acquired by a company called Recursion, publicly traded company with a lot of cash. There is a company called Benevolent AI in the UK. They just fired 180 people, closed the lab, also announced in a very kind of gentle way that they flopped in phase 2 because of lack of efficacy, did not demonstrate efficacy, and they are a target discovery company, so they position themselves as a really, really professional target hunter. So in theory, if you fail on a target choice, that's not pretty, and they are suffering, and many others. I get probably 1 to 2 calls a week about bias, alarm, you probably need to actually hire somebody. Now there are 3, 4 companies on the market that are very solid, that have demonstrated success. One of those companies I can openly talk about is Schrödinger, that we respect. This is a computational company from the 90s. They were selling software. Now they also started developing their own drugs. I don't think that they are as fast in drug discovery as we are. And also they usually go after very old targets, established targets that are announced. So the level of novelty may be not as high, but it's a very good company. Publicly traded, so no financial advice, no forward-looking statements, etc. So don't buy or sell them based on this interview. But we respect these guys because they also sell software and enable the market. So at least you see that it might not be AI, but it's credible computational tools. There are a few others that are okay, but we have not seen them selling software. And what Insilico does, it's super transparent, because if you do have the money, you can actually acquire our capabilities and buy PandaOmics, Chemistry 42, Insilico, so huge user base. Hundreds of key opinion leaders are using PandaOmics right now. Many pharma companies have deployed Chemistry 42. And now a new company which can develop maybe some fancy model in chemistry and start screaming about it, pharma companies would say, oh, okay, let's do a benchmarking study with Insilico. And usually they don't agree to that.
Just to say that PandaOmics, Chemistry 42, and Insilico are the 3 stages of your AI process.
That's correct. So we actually rebranded them a little bit. So now our platform is called Pharma AI. Underneath, we have 3 building blocks, 3 platforms. One is called Biology 42, Chemistry 42, and Medicine 42. And underneath, we have a bunch of apps. The main app in Biology 42 is called PandaOmics. That's used for target discovery. A lot of key opinion leaders use it. Many academic institutions, also in the UK. It's so easy to use that high school students can use it, and some of them do, and some of them publish great work. Another tool is called Chemistry 42. It's only deployed in big pharma because it's very expensive. It's also very expensive to set up and run, so you need to have supercomputing infrastructure to do that. And we usually deploy that on their private cloud or their supercomputer. And in Insilico, it predicts the outcomes of clinical trials. So phase 2 to phase 3 transitions, we validated over 7.5 years. We have got a paper coming up explaining how we did it. And now this tool is being piloted predominantly by hedge funds and banks, believe it or not pharma, because they are trading based on the tool.
So what I'm hearing is that Insilico has a variety of assets. It has deep technical understanding of AI, but also biochemistry. And you've got this approach in which you can open your platform to many users. But it seems there's even more coming along the pipeline. I have seen news recently of some potential breakthroughs with quantum computing as well as traditional computing. Wonder if you can say maybe just a minute or 2 on what this involves.
Sure. So I personally believe that quantum computing is going to change the world in the next 5 years. Most likely even in 2 years. Because IBM, unlike with Watson, they have hyped it up quite a bit. You remember they were hyping up AI quite crazily without really good use cases, even though now you can see that those claims have been superseded by ChatGPT and other tools. They were just too early. But with quantum, IBM is actually pretty silent. But you can see if you are a user, now you can actually get access to a 50-qubit machine and use it. And so companies like ours can actually apply, get access, use it. Now they have over 100 qubits, and now they're rolling out massively scalable systems. Microsoft just announced their quantum strategy, and that's not just for babies. I've seen some of that in action. It's the real deal, so maybe they're undervalued even at their crazy valuation right now because they're such an investor in quantum. And Google is also doing great work. So what we decided to do, we decided to look at our generative chemistry engine and think, okay, well, can it be accelerated by quantum? And we realized that maybe our generative chemistry engine is the lowest hanging fruit for the quantum computing companies to actually build use cases around, because both the generation step and the reinforcement learning step can be dramatically accelerated using quantum. And as I've explained to you in the beginning, we are not exactly interested in super high accuracy when doing snappy generation. We are interested in getting diverse, novel structures. Very often they will go way outside known chemistry data sets and libraries. So we want that. And the most important part is to ensure that the reinforcement learning system, the system that is evaluating the quality of those molecules, also does a great job and doesn't let the bad ones pass. Both steps can be accelerated using quantum. Both steps can accept a little bit of error rates, so the error rate can be there. And we've published one paper recently showing the quantum supremacy with one quantum model.
How many qubits in that?
So for that one, we did not really run it on a real quantum computer, we simulated. So now we have published another paper which demonstrates how the many different parts of chemistry 42 can be accelerated by quantum. And of course, usually, if you know how I do academic research or academic publishing, we usually have a few papers that explain what we do, and then we publish an experimental paper on a real quantum computer, usually with experimental evidence, with synthesis, testing, etc. Currently, that's in the works. And as they say in the Middle East, where I spend quite considerable time now, inshallah, maybe this year, maybe early next year, we'll be able to demonstrate the quantum-generated molecules on a real quantum computer, and preferably not on just one quantum computer, but on several, just to demonstrate that now a production-grade generative chemistry system can be run on a quantum computer. So that's my dream. I think that I will be able to realize that. Now the company has readjusted its management structure as well. So one of the reasons why we were quite successful in drug discovery and development was because I gave most of the throne at Insilico to a super experienced drug hunter, PhD from Harvard, 11 years at GSK, and then he listed a huge contract research organization with thousands of scientists doing chemistry and biology for the others, and took it to billions of dollars in market cap, and then joined Insilico. Now he is responsible for probably 80% of the company resources using AI to discover drugs. And now I can spend a little bit more time and resources to do robotics, quantum, and generative. We also just released generative biologics.
Fascinating stuff. And as they also say in the Middle East, mahaba. We're running out of time, Alex, but I do want to just quickly ask you about longevity, because I know that is the underlying dynamic force that drives you, and you're well known as being an absolute workaholic and dedicated to extending human life. When we last spoke to you, you had become a little bit pessimistic. Your timelines for longevity, escape velocity, the time when science gives you back a year every year that passes, was slipping further into the future, which for people like David and myself at our age is extremely bad news. Are you getting any more cheerful, or is it still quite some way in the future in your view?
Well, I definitely now know that with pharmacological means, we don't have a chance to achieve longevity escape velocity. We probably can win sometime, because now I know how difficult it is to discover a drug and how marginal the effect is going to be on longevity. But those pharmacological means provide us with the tools to study aging much more deeply and understand human biology much better. So now I'm a little bit more optimistic because of consumerization of GPTs, for example. We just published Precious One GPT, which utilizes methylation and transcriptomic data for aging clocks, and at the same time learns the fundamental biology of several diseases, age-associated diseases, and you can now query it for protein targets that are implicated in aging and disease at the same time. So if you want to look it up, just Precious One GPT, one clock to rule them all, so to speak. And we are going to be doing many more of those this year. So our fundamental understanding of human aging biology and interpretation of that into actionable targets has increased. However, I don't want to give you any estimates. I don't like to talk about forecast into the future. I think that regardless of what you do, set your longevity expectations to 120, 150 anyway. It's good for you, just for your mindset, regardless of whether it's going to happen or not. Just from the mindset perspective, you need to be prepared. However, as a scientist working in the pharmaceutical industry, I do not want to give any estimates. I just want to say that the future is great. It's going to be even better. But now we also have many other factors like Neanderthal with a huge baseball bat. I'm talking about some rulers of totalitarian countries with the red button that they like to talk about nowadays, and they might actually push it. And then we need to think about the longevity bunker where we are going to continue the research. You know, the risks associated with geopolitical tensions are real. And I hope that people will wake up and start thinking about longevity more than they think about skin color, their ethnicity. And for some strange reason, their country is better than another country. You know, we're all human and we're all going to die right now. Well, not right now, but soon. So we should wake up and think about that more than about some variable values that people have. The most fundamental value is human life. Unless we see more people realizing that, there will be always a huge risk.
That is a very good way to end it, Alex. Alex, as before, as always, an absolute pleasure. Thank you very much. A fascinating conversation.
Thank you so much.
I really look forward to welcoming you back when you can tell us the results of the phase 2 trial. But I'll bet you've got some other exciting news before then anyway.
Please welcome to ARDD. This year it's the 10th annual Aging Pharma Conference, largest conference on longevity biotechnology. So if any of your listeners want to learn about aging research, 5-day event, everybody in aging is there, epic location.
Where is it taking place?
It's Copenhagen.
Copenhagen.
Every year, same place. University of Copenhagen is hosting, nonprofit. I'm sponsoring myself. Sponsoring is Insilico. Boston Consulting Group is a knowledge partner. End of August, beginning of September. That is August 28th to September 1st. 5 days. Longevity Medicine Day is there as well. So many doctors are going to be there. Look it up. It's the most epic event. Last year, we've got 450 on site, about 2,500 online. David Sinclair is there, Vadim Gladyshev, Eric Verdon, Nir Barzilai, so the entire crew.
We'll put the information in the show notes. And I look forward to getting out of longevity bunkers and into the longevity dividend as soon as possible.
Thank you very much for coming back on the podcast, Alex.
Thank you.
Thanks, Alex.